micro nano education for everyone.
First, we're going to have two presentations. Dr. Neda Habibi from the University of North Texas and Dr. Darsh from Louisiana Tech. We're going to have Neda go first. So Neda is a member of the MNT-EC community, active in the community. If you go to some of our professional development sites, you'll probably see videos of her explaining student presentations. And she was previously with, what was your prior institution?
Northwest Vista College.
Yes, Northwest Vista College.
NVC.
Yes, NVC. And is now with North Tech. So we're excited to have her back here in the capacity and presenting formally to the MNT CURN community. So Neda, take it away.
Thank you, Kendrick. So my name is Neda Habibi. I'm assistant professor at the Department of Biomedical Engineering in University of North Texas. Before joining UNT, I was a coordinator with a nanotechnology program at Northwest Vista College. It's a community college in San Antonio. Again, it's in Texas. And also, I was a faculty at UT San Antonio. I actually got my PhD in nanotechnology from University of Genova in Italy, and my research is focused on integrating nanotechnology, nanobiotechnology with biomaterials, specifically for drug delivery purposes, which I'm going to explain a bit about this today. And when I was at NVC Northwest Vista College, I got in contact with Jared, with also the Micro Nanotechnology Education Center. I've been working with this center from the start. And we have been working on the journal, establishing an academic journal, providing curriculum, and also with this undergraduate research projects. So my research is focused on using nanobiotechnology and nanomedicine, especially for drug delivery application and also cancer treatment. So I'm going to explain about my research and about the projects that you will be, students will be doing if they are hosted by the UNT. So first of all, I'm going to give you an introduction about drug delivery with nanomedicine. What is nanomedicine? Why do we need nanomedicine? What's the problem with conventional drug formulation? And I'm going to talk about briefly talking about a couple of my research projects, which is, for example, making microcapsules, polymeric capsules, making self-assembling peptides and proteins, and also making nanofibers and nanotubes. We know that nanotechnology is the science and technology of working with small nanoscale material in the dimension of 1 to 100 nanometer. Although this is a very new technology, it has been revolutionized over the past 30 or 40 years, but we now know that nanoparticles have been made over hundreds of years ago. One famous example is the Lysurgus cup, which is from the 1st century. Gold nanoparticles are used in this cup. So we know that nanoparticles were made hundreds of years ago. We just didn't know that they were existing until recently that we have developed techniques, spectroscopy and microscopy technique. Scientists have developed these techniques. So now we are able to see them, characterize them, and use them in devices. And what you see here is an atomic force microscopy that was invented in 1986. So nanobiotechnology is also a very new area, is an integration of nanotechnology and biotechnology. We don't have like a specific definition and term for that. For example, Nature has defined nanobiotechnology as using the tools from nanotechnology for studying biological phenomena. For example, sensors, for example, the cell membranes. But also the Nanobiotechnology Journal has defined nanobiotechnology as nanoscale systems for making smart nanoparticles and nanobiomaterials. And by smart, we mean that we want to upgrade the properties of biomaterials and make them better. But just one important thing here is that we have a lot of natural nanoscale biomaterials, like, for example, DNA or protein. But just simply studying DNA and protein doesn't mean that we are using nanobiotechnology. We have to use nanotechnology tools to modify those molecules, make them better so we can use them in different devices. In nanobiotechnology, mainly we want to use them for biomedical engineering purposes. So nanomedicine is also a new area, is application of nanobiotechnology in medicine. NIH has defined nanomedicine as highly specific medical intervention at the molecular scale for curing diseases, for example, making new nanomedicine for repairing damaged tissues, tissue regeneration, like, for example, bone, muscles, or nerve. Anything in nanoscale that is also related to health, for example, devices, nanoparticles, in nanoscale that are also related to health could be in the category of nanomedicine. So when I talk about nanomedicine, you might quickly think about pharmaceutical, new medicine, which is also correct. But nanomedicine is not only nanopharmaceutical, it's about any kind of device or particle in the nanoscale that is related to health. It could be in the category of nanomedicine. For example, we have nanodiagnostics, biosensors, imaging structures that are used for detecting molecules. One important example is magnetic nanoparticles and gold nanoparticles. We have nanopharmaceuticals, for example, making microcapsules, making nanoparticles that can be used for delivering drugs. In the area of regenerative medicine, as you see in this picture, we are mainly making scaffolds and nanofibers that can support cell growth outside human body, and we can use that as mimicking the behavior of extracellular matrix and providing that scaffold with the cell and using that for repairing damaged tissues. With implants, we have nanocoated stents that are used in patients with coronary artery diseases. We have nanosensors. So all of these, we are using a device or nanostructure that is related to health, and they are all in the category of nanomedicine. So why do we need nanomedicine? What's the problem with conventional drug formulation? So first of all, when we are administering a drug that is in a free form, is not modified with nanoparticles, is not modified with nanostructures, this drug is going to be distributed inside our body in a free way. So what's going to happen is that the concentration of this drug, as you see in this diagram, is going to get increased, which is usually above the toxicity level of that drug. So what is happening is that we are having some side effects on normal tissues. And then quickly it's going to get eliminated by organs such as liver and kidney. So as you see in this diagram, the concentration is going to get decreased. So we have to repeat the dosage to have the same therapeutic effect. So what is happening, we are having side effects and then decreasing the concentration. We have to repeat that dosage over and over again. So we are having also some drug overdose. Also, another thing that's happening is that once this drug is getting distributed in a free form, it's targeting normal and healthy tissues, as well as the target tissue, which could be a cancerous tissue. So this is not advantageous. What we want is we want to control the release of drug inside our body. So simply by protecting them with a capsule, with microcapsules, with polymers and nanoparticles, we can protect and control the release of drug. We want to decrease the concentration of the drug to a lower level, so they are below the toxicity level, and keep them for a longer period of time. So this is the overall goal, that we need to modify conventional drug formulations. So how nanotechnology can actually help us to overcome these problems? First of all, we can make molecular carriers. For example, we can make capsules, we can make polymers, we can make nanotubes to host and load the drugs and control the release of these drugs. With nanomedicine, with nanotechnology, we can modify the surface of these carriers. For example, we can add targeting ligands, molecules that can identify specific cells. We can increase targeting functions. We can improve the stability, especially for one important example is chemotherapy drugs. They are hydrophobic. They have low solubility in water. So just making nanoparticles of a chemotherapy drug can increase the stability and also solubility of these kind of drugs. We can improve cellular uptake. So if we have a smaller particle, a nanoparticle that can enter a cell membrane easier and can have better therapeutic effect. Overall, we want to add some smart activities. For example, adding ligands that those molecules, those drug molecules can actually identify targeted tissues. For example, if that target tissue is a cancerous tissue. Overall, we want to decrease the side effects and we want to increase the therapeutic effect of drug molecule. Especially in my case, I'm using anti-cancer drugs for chemotherapy treatments. So a couple of the research projects that we are doing at UNT will be developing microcapsules from biomaterials, making self-assembling proteins, and also making nanofibers using electrospinning technique that I'm going to explain all of these briefly. So the first one is making microcapsules using a technique which is called layer by layer self-assembly. So layer by layer self-assembly is a spontaneous formation of organized structures without any external force or without any chemical reactions. Molecules can self-assemble themselves into organized structures. Especially the LBL, this is called the LBL self-assembly, is a very simple and versatile technique. We can use this technique to make ultra-thin films. And also having programmable properties, meaning that we can adjust the properties of these capsules in the way that we want. So how this technique works is a very simple technique. We just need two polymers that are charged. So the LBL works on alternate absorption of opposite charged molecules. So we need a polymer that is negative charge, a polymer that is positive charge, and if we have this alternate absorption on a flat substrate, we get multi-layers, ultra-thin films. We can also do this process on a spherical particle. For example, having a sphere, putting those polymers on top of this sphere, we can make a shell and macrocapsule out of these microspheres. So in this picture is an electron microscopy image of these capsules. We can also make them empty. So once we make these capsules, we can load them with a drug, and we can also use that for having controlled release of drugs from these capsules. And we can also test them against cells. So what we need to make these capsules, this is going to be one of the projects that you will be working if you're hosted at UNT. As I said, this is a very simple process. We only need a microsphere, which we can use carbonate calcium. The only thing that we need charged microsphere. And also we need biopolymers. Because we are using these capsules in human body, we don't want to use synthetic polymers. We want to choose a biopolymer so it doesn't have toxicity effect on cell. So a lot of biopolymers that we can choose are chitosan, collagen, alginate, even proteins. As long as they are charged, we can use them for making these capsules. And also we need some diluted form of acid to make these capsules empty. So we get these microspheres with these charged polymers. For example, we are starting with a negative charge polymer. We make one layer on top of these microspheres, and then it's just repeating through this process until we have a fine multi-layer shell on these coated spheres. So we have alternate absorption of negative and positive charge polymers. We are making step by step with the self-assembly process. This is a pure self-assembly process. We are making these layers on top of these spheres. And at the end, once we have a good thickness of these nanolayers, we can delete the core using some diluted form of acid, and we can have an empty capsule. This is a very, very interesting process for making microcapsules and using them to deliver drugs. So these microcapsules can be used for loading drug molecules, for example, anti-cancer drugs. They are used for controlling the release of drugs. We can also add some ligands at the surface of these capsules to make them smart. For example, they can identify cancerous tissues and attach to them, target these cancerous tissues specifically. So we want to decrease the side effect of chemotherapy drugs. This is just one example that we are coating these capsules with a protein. So we can add some ligands on these proteins and make these capsules more smart. So what we will be doing as a research project in the nanomedicine lab at UNT, we will make these capsules, we will load them with the drug. So we have to characterize these capsules in terms of charge, size. We want to know how much these capsules are able to host the drug and also how much they can release the drug and what kind of triggers we can use to release drug molecules from these capsules. So another thing that we will be doing is using the cross-crystal microbalance. This is a very interesting instrument to measure the multi-layered thin film growth. So QCM or cross-crystal microbalance is used based on quartz sensor, which is piezoelectric. And this as you can see here, this is a gold sensor and also quartz sensor. And these are all piezoelectric, so meaning that once we have a mass deposited on the surface of this quartz, we are creating a resonance frequency. As the mass is getting deposited, this oscillation is going to change. So this oscillation is correlated to how much mass we have on the quartz sensor and what is the thickness of these multilayers. So as I mentioned, the shell of these capsules are in nanoscale. So if we want to monitor how we are making these shells, what is the thickness, what kind of mass we have in each step, are we actually having those polymers absorbed? We can use the quartz crystal microbalance to kind of monitor what we have on the shell of these capsules, especially if we want to use a drug inside the shell or inside the capsules. We can use QCM to monitor multi-layer growth. So another thing that we will be doing in the research project is using these capsules for delivering drugs and kind of closing these capsules and opening them once we want for in order to deliver the drug. So the question is, once these microcapsules are delivering the drugs and they are reaching the site of disease, how they can actually get open and how they can release their content. So one interesting thing about these capsules is that they can have a closed and open state using external triggers. For example, changing the pH, because these layers are all polymers. They have ionic charge. And once you alter that ionic charge, it's going to make that open. And this is reversible. You can close it. So once these capsules are reaching the site, you can change the pH. Especially for tumors that have low pH, you can open them. These drug molecules will go out and then they can get closed again. Also for loading drug molecules. So we will be characterizing these capsules in terms of what triggers we can use to kind of open and close them. So another project is working on self-assembling peptides and proteins. And this is actually these are very interesting molecules that have self-assembling capability. So as I mentioned, self-assembly is a spontaneous process of making organized structures. The driving force for self-assembly is intermolecular forces. For example, hydrophilic interaction, hydrophobic interaction, van der Waals interaction. All of these will allow a molecule to stick together and make nanotubes and nanostructures spontaneously. So the molecule that we will be using is a peptide. I think you all know that peptides are made of amino acids. Amino acids do have amine groups and also carboxylic groups. For this specific peptides, we also have a aromatic ring as the structure of these amino acids. So these aromatic rings can actually have hydrophobic interaction and they can stick together. And so these molecules can stick together to make nanostructures. And this is what we will be using to make nanotubes. So the molecule that we use is a dipeptide. It's called diphenylalanine. I will call it FF. This is a very short sequence of peptide. It's a very small molecule, but it can spontaneously make nanostructures. And also it's interesting to know that FF is associated with Alzheimer's disease. So patients that have Alzheimer's disease, a beta amyloid polypeptide is formed in their brain that this FF is associated with making development of these polypeptide. So in this picture, you see once we have this 1 unit of FF, 6 of them will stick together to make a hexamer. And then these hexamers will stack together and we will have narrow channels. And these narrow channels will also stick together in the process of this self-assembly to make nanotubes. And nanotubes will also stick together to make bundles of microstructure. So with a pure self-assembly mechanism, we are making nanotubes. The goal is to use these nanotubes for loading drug molecules, such as anti-cancer drugs. So in this research project, we will use the FF structure with pure self-assembly mechanism. We will make nanotubes from these. We will load them with the drug. The drug that we are using is called flufenamic acid and also fluorouracil, which is an anti-cancer drug. Especially these two types of drugs are hydrophobic, so we want to know how they are loaded inside these nanotubes and how they are released from these nanotubes. So remember, the overall goal is to extend the release and keeping these drugs with lower concentration for an extended period of time. So another project that we will be working on at UNT is using electrospinning to make nanofibers. Electrospinning is also a very interesting technique to make nanofibers. I don't know if you're familiar with this technique, but I'm just going to briefly explain. In this technique, we have a needle, we have a polymer, electrospinnable polymer. We actually apply a voltage between the needle and the collector should be also conductive. Once we are applying a high voltage, the polymers that are ejecting from this needle will get deformed as a result of these repulsion forces, and they will produce a deformed droplet called Taylor cone. And at a certain high voltage, these repulsions are overcoming the interactions between these molecules, and a jet will be ejected from the needle, which is collected on the fiber as collected on this substrate as nanofibers. So in electrospinning, we have the needle, we have the high voltage and the collector, and using appropriate type of polymers. We can also use biopolymers such as chitosan and collagen. We can have a scaffold of fibers. We can use these scaffolds for different purposes. For example, drug delivery application, and also using these scaffolds for supporting cell growth. So in one of the projects, we use these. This is actually a picture of electrospun nanofibers. These are chitosan nanofibers. And what you see, we have modified these nanofibers using nanoparticles, such as magnetic nanoparticle. And this is a very interesting experiment that once we expose these fibers to an alternate magnetic field, it actually can break these capsules. So as I said, one of the questions is how we can break our structures to release the drug molecule. So using external triggers such as magnetic nanoparticle is one way to break these capsules, break these fibers, and release the drug molecules. And in this picture, you can see how we are how we made these polymers and nanofibers and use magnetic triggers to break these fibers. And also future projects that we can work on, we can make hydrogels from these peptides and proteins with also this pure self-assembly mechanism. We can use these hydrogels to load different kind of drugs, for example, anti-cancer drugs. Also, one interesting application is using for local chemotherapy gels. If we want to locally deliver chemotherapy drugs, these peptides and proteins can make hydrogels, as you can see in this picture, hydrogels that can encapsulate chemotherapy drugs and release them locally to the site of disease. So these were just a brief explanation of the research, some of the research projects that we will be doing at the Nanomedicine Lab at UNT. And these are all focused on using nanotechnology for making nanomedicine, new drug formulation, and scaffolds of cell. So I'm going to end this talk here and open it to any questions that you might have. Also, if you have any questions, you can also email me anytime you want and ask your questions.
All right. Well, Dr. Habibi, in 2018, I was diagnosed with epilepsy. It took me about 2.5 years for my, medications for my body to adjust the medications. And I still get, this aura of, that's the feeling you get prior to a seizure. So the medicine, what I take regularly, isn't as effective. Now, you mentioned microcapsules. It sounds like, would they be able, like, let's say they're, they go to a part of my brain or something, would they be able to sense, let's say, an aura or, such a small change in my, brain activity that could lead to an actual seizure? Because during those 2.5 years, I couldn't go anywhere. Well, I couldn't drive, is what I mean to say. So would that be an application of microcapsules? So
thanks for the very good question. First of all, I'm very sorry to hear that you had this problem.
Oh, no.
And I hope you will get very better soon. So actually, for delivering drugs to the brain, one important hurdle is the brain-blood barrier, which this is a challenge for delivering drugs to the brain. Although this is not my expertise, but the BBB membrane is actually the hurdle for delivering drugs to brain. And so in this area, I know that as the medicine gets smaller, for example, nanoparticle, they can escape these brain-blood barrier and get to the brain and do their effect. So this is going to be the effect of using, for example, nano-sized medicine in terms of like bigger medicine, for example, microstructures. So for these microcapsules, they are usually like bigger type of structures in the microscale. But I would say, although this is not my expertise, but using nano type of capsules, nanoscale capsules, and nanoparticles would be one way to overcome this hurdle, which is delivering drugs, passing those drugs from the brain-blood barrier, which is actually a membrane that is will diffuse all of these molecules and will not allow permeation of larger molecules to the brain. So I think if we have smaller type of nanocapsules instead of microcapsules or nanoparticles, they will work better for delivering a medicine to the brain. So I hope that answers your question.
Yes, absolutely. I actually have one more question.
Sure.
You mentioned the blood-brain barrier and, using smaller, what was it you call them? Smaller particles?
Yes, nanoparticles.
Nanoparticles. Now, I was reading this article about xenobots, which are apparently the world's first living robots, and they were actually assembled from heart and skin stem cells. I believe it belonged to some kind of African clawed frog or something like that. And apparently they can move independently throughout the body for about a week before running out of energy. And they can actually self-heal as well as break down naturally. Could those nanoparticles be used in conjunction to those xenobots?
Can you explain again what these robots are made of?
Yes, let me just type that here. All right, so apparently they are, not apparently, but they are these xenobots, and they're the world's first living robots. And they are also self-replicating, and they were assembled from the heart and skin stem cells. I believe it belonged to an African frog.
Okay.
And from what I read, from what I saw, they can actually move independently in some kind of host body for about a week before running out of energy and breaking down naturally.
So that's also a very interesting thing. I've also heard about like small nanorobots. Again, this is not on my expertise, but what I know about nanorobots is that so they need an external trigger to be able to have some kind of locomotion. And that's why we call them, like, for example, robots, or robots that can be injected and move inside our body. So that external trigger could be many things. For example, magnetic nanoparticles, if we use them, we can use a trigger that provides locomotion. Or, for example, gold nanoparticles that are exposed to light, they can also provide some kind of locomotion. So in this area, we need to have locomotion from these particles, so we are able to name them robots. So what you said, I think these xenobots are made from stem cells. I don't know how they are able to move inside the body. What's the trigger for them to move inside our body? Right. So that's interesting for me to know. If you want to put that link in the chat box, I can take a look at that. And I've also heard about nanorobots, but sometimes they call it a robot, but it's actually not a robot. So you have to make sure how they are moving inside the body. What is the trigger that they can actually move? So thanks for the question. That's also interesting.
Awesome. Thank you for the answer as well. Thank you.
You're welcome.
Thank you so much.
You're welcome.